Welcome to the NCOH monthly webinar of January 2021. I am Martin Hoek, Communication Manager for NCOH and your host for today. This meeting will be recorded and made available through the NCOH website. This month we'll have presentations on avian influenza, also known as avian flu or bird flu. Avian influenza was first detected in 1878, if I'm not mistaken. Before I give the word to our guest today, just a reminder that you can ask questions using the chat function. So let us start with the presentations. Our first speaker today is Professor Thijs Kuiken. He is Professor of Comparative Pathology at the Department of Virus Science of the Erasmus University Medical Center in Rotterdam. His work focuses on research on emerging viral diseases such as avian influenza and MERS using a One Health approach. He is also coordinator of various projects such as Zoonosis in the Night, which aims to evaluate the potential of viruses in bats to spread to people and domestic animals, Delta flu, to determine the key factors determining the spread of avian influenza between poultry and wild birds, and Antigone, a European project investigating the underlying causes for the emergence of infectious diseases from animal reservoirs to humans. Today, the title of Professor Thijs Kuiken's presentation is One Health Evaluation of Highly Pathogenic Avian Influenza in the Netherlands, 2020/2021. So don't forget to ask questions using the chat function. And then for now, Professor Thijs Kuiken, the floor is all yours.
Thank you very much, Maarten. And thank you for this opportunity to discuss high-path avian influenza from a One Health perspective. Right at the beginning, I acknowledge both the funding agencies and the people who have made this research possible. I've highlighted Valentina Caliendo, who is working on this outbreak as part of her PhD training. The subject of this seminar is the current outbreak of high-path avian influenza in Europe, including in the Netherlands. And you can see here a figure of the outbreak by the European Union Reference Laboratory, showing the poultry farms as round dots and the wild birds as squares that have been diagnosed positive for high-path avian influenza since last autumn. So, how come Europe's poultry industry is not protected from this disease? What do wild birds have to do with it? And how can we improve the situation? To answer these questions, we need to take a broader view, both in space and in time. At the same time as this high-path avian influenza epidemic, there's a more serious and more complicated crisis going on. And I don't mean COVID-19, however devastating that is. I mean the global ecological crisis, because human society is living beyond Earth's means. As you can see in this figure, we are already living beyond the safe operating spaces of climate change, biodiversity, eutrophication, and land use change. According to research over several decades by many scientists, a radical transformation of human society is required, defined by IPBES as a fundamental, system-wide reorganization across technological, economic, and social factors, including paradigms, goals, and values. And in my view, all applied scientists should keep this in mind in their work. And in the field of biomedical sciences, including influenza research, the One Health approach is a suitable way to do so. The One Health approach has gained popularity in the last 20 years as an appropriate way to deal with health issues that cross the boundaries between humans, animals, and the environment. There are slightly different definitions of One Health in circulation, depending on the background of the authors. A widely used definition by the American Veterinary Medical Association is the integrative effort of multiple disciplines working locally, nationally, and globally to attain optimal health for people, animals, and the environment. And in this presentation, I'll assess the One Health impact of both high-path avian influenza and the poultry production systems with which it is so closely linked. To understand the emergence of high-path avian influenza, we need to know something about the history of poultry production and consumption. In the last 60 years, the demand for meat and eggs in the European Union has increased. And here's a graph showing the per capita consumption of meat from different food animals and per capita egg consumption. The increase in egg consumption is not so high, only 1.1-fold. However, the per capita consumption of poultry meat, mainly from broiler chickens, has increased a lot, more than 4-fold. In part, this is due to effective marketing. In a review from the 1980s, it is stated, aggressive marketing methods increased the per capita consumption of broilers more than 5-fold in the 3 decades beginning in 1950, and further substantial increases are predicted for the future. Such an increase in demand requires a parallel increase in production. And that is indeed the case. In the same period, poultry meat production in the EU increased 6.7-fold and egg production increased 1.6-fold. If we focus on poultry production in the Netherlands, there's a similar increase in egg production, 2.1-fold. However, the increase in poultry meat production is nearly twice as high, 12-fold instead of 6.7-fold. An important reason for this rapid increase is due to a quirk in an international trade agreement in 1962, the so-called Dillon round of the GATT negotiations. In these negotiations, the EU agreed to low or even zero tariffs for imported cereal substitutes, such as soybeans, resulting in a massive import via Rotterdam Harbor, the so-called Gap of Rotterdam, or in Dutch, Gat van Rotterdam, in the EU trade wall. These cheap cereal substitutes made it highly profitable to raise poultry in the Netherlands. So the reason is therefore not because Dutch people eat more chicken than other Europeans. Less than 30% of poultry meat produced in the Netherlands is consumed here. The majority is exported to other European countries, in particular Germany and the United Kingdom, which have relatively small poultry industries. In the period that the Dutch poultry industry grew so much, it also intensified and consolidated. The number of poultry farms has decreased from more than 300,000 farms in the 1950s to less than 2,000 farms nowadays, with an average of 80,000 broilers and 40,000 layers per farm. In 2015, the poultry industry had an added value of 1.46 billion euros to the gross domestic product of the Netherlands. And in total, 22,000 people are employed in the poultry industry, most of them in meat processing and in the supply of feed, stables, and mechanical installations. Because of the large degree of mechanization, the total number of poultry farmers in the Netherlands is a few thousand. And in any health policy changes, the livelihoods of these people need to be considered. On the downside, the intensive poultry industry in the Netherlands has negative health impacts for people and the environment, recently analyzed together with other livestock systems by Imke de Boer at Wageningen University and her colleagues. Human health impacts consist of disease from inhalation of fine particulate matter, zoonoses, antimicrobial resistance, odor annoyance, and industrial accidents. Environmental health impacts consist of soil and water pollution, especially by nitrogen and nitrates, greenhouse gas emissions, and use of land and fresh water. I'd like to highlight just a few of these. Regarding human health, the emission of particulate matter from skin, feathers of poultry, from litter, feed, and manure is concentrated around poultry farms, causing premature death and several respiratory and cardiovascular diseases in local inhabitants. The estimated impact is around 1,900 disability-adjusted life years, or DALYs. Known zoonoses, particularly Campylobacter infection, have an estimated impact of around 1,300 DALYs via direct contact or via the environment. Emerging zoonoses, like high-path avian influenza, are considered somewhat probable in poultry flocks and have a severe potential impact. Regarding environmental health, the estimated greenhouse gas emission from the poultry industry is about 9 million tons of CO2 equivalent. And if we consider that an average Dutch citizen has an emission of about 15 tons of CO2 equivalent per year, this corresponds to the annual emission of about 600,000 people, which is about the size of the population of Rotterdam. The estimated land use by the poultry industry, mainly for producing poultry feed in way of cereal and protein-rich crops, is 8,400 square kilometers, which corresponds to about 1/4 of the total land area of the Netherlands. In summary, the current intensive level of poultry production has substantial impacts on human and environmental health. The intensification of the poultry industry also has increased the risk of high-path avian influenza virus infection. This virus is a mutation from so-called low-path avian influenza virus, a virus that occurs in wild water birds and causes little or no disease. Out of 37 conversion events to high-path avian influenza virus recorded worldwide between 1959 and 2015, 34 occurred in intensive poultry production systems, mostly in Europe. And therefore, intensive poultry farms are the chief source of highly pathogenic avian influenza virus. This virus has a marked impact on poultry health. By itself, the virus already causes high mortality among poultry, at least in chickens and in turkeys. Furthermore, the current European Community rules are to kill all birds in a commercial poultry flock where high-path avian influenza has been detected, so-called culling. By carrying out this measure, it's permitted under European rules to start raising poultry and exporting poultry products again after only 3 months. So far during the current outbreak, the virus has been detected on commercial poultry farms at 11 locations in the Netherlands, and over 500,000 birds have been culled. An even larger outbreak occurred in the Netherlands in 2003 and spread to the most poultry-dense areas in the Netherlands, the Gelder Valley, shown here, and South Limburg, as well as to Belgium and to Germany. During that outbreak, 30 million birds, that's about 1/3 of the total poultry population in the Netherlands, were culled. Arjan Stegeman and colleagues from Utrecht University analyzed this outbreak and concluded that current measures for influenza control are insufficient to stop such an outbreak happening again. And to prevent future outbreaks, they suggested it might be worthwhile to consider reducing the flock density of commercial flocks, or to consider vaccinating poultry as an additional control measure. However, neither of these measures have been implemented to any degree, and both areas are still at high risk of such an epidemic. A relatively new phenomenon in the epidemiology of highly pathogenic avian influenza is its spread by wild birds. Until 2005, this was not known to occur. However, since then, high-path avian influenza virus has spilled over from infected poultry to wild birds several times. Some wild birds suffer high mortality and are therefore victims of the infection. Other wild birds are able to carry the virus over long distances during autumn and spring migration, as you can see on this map of the globe, and therefore act as vectors of the virus to other continents. In this way, high-path avian influenza virus, the subtype H5, has spread repeatedly from Asia to Europe in the last 7 years. causing disease in both poultry and wild birds. The biggest outbreak ever recorded in Europe was in the winter of 2016-2017, as you can see on the right, when 1,200 poultry holdings in 24 countries were infected, and the disease was recorded in wild birds in 1,500 events, involving 70 different species of wild birds in 29 countries. Although we know that wild birds are both vectors and victims of high-path avian influenza virus, we have a very poor idea of the actual scale of wild bird mortality from the disease. We first attempted to estimate this impact in the Netherlands during the 2016-17 outbreak. When the outbreak already had begun, we started requesting mortality data from organizations gathering such reports or that were removing carcasses. In this way, we counted 13,600 wild birds of 71 species that were reported dead. However, we estimated that the actual mortality was between 10 times higher, between 4 to 10 times higher. As you can see in this graph, the two main species found dead were tufted ducks and Eurasian widgeons. Besides these two species, great black-backed gulls and peregrine falcons also may have suffered substantial population declines. Unfortunately, half of the carcasses, shown in light gray in this figure, were not identified to species. In the last few months, starting in October 2020, there's been another incursion of high-path avian influenza in the Netherlands and elsewhere in Europe. The features of this outbreak in wild birds differ somewhat from previous outbreaks. First, high mortality has been seen in unusual species. On the German Waddensee coast, over 10,000 wild birds were found dead, 75% of which were either barnacle geese or Eurasian widgeons. On the Dutch Waddensee coast, the birds found dead most often were barnacle geese, a species in which high-path avian influenza has not been reported in previous years. Another novel species was the red knot, a species of wader, about 1,500 of which were found dead or ill on a single beach of the German Waddensee, and which tested positive for high-path avian influenza virus. Small numbers of affected red knots also were found further south in Germany and on the coast of the UK, on the other side of the North Sea. Second, neurological signs were often observed in affected birds, and the links that are typed in here show examples of these nervous signs in a barnacle goose and in a red knot. Third, a high prevalence of infection was found in wild duck species, for example, mallards, Eurasian widgeons, and common teals that were either trapped alive or hunted without any clinical signs of disease. And this was done in the Netherlands, in Germany, and in Italy. And in fact, in Italy, most of the high-path avian influenza virus infections involve asymptomatic wild birds. Very early in the outbreak, we set up a consortium, AI Impact 2021, including many Dutch organizations involved in nature management, ornithology, and disease diagnosis. Our primary aim is to estimate the level of wild bird mortality from high-path avian influenza as a function of population size. And additional aims are to relate the mortality in wild birds to high-path avian influenza incursion into poultry farms and to identify the range of species affected by high-path avian influenza in the Netherlands. Every week we publish maps on the website of the NVWA showing where in the Netherlands wild birds have been reported dead. The maps show 4 different categories of birds: ducks, geese, and swans in the first place, then raptors, then other water birds, and then other terrestrial birds. Increased numbers of dead wild birds were reported from the middle of October to the middle of December, about 5,500 in total, with a concentration along the coasts of the Dutch Waddensee, although wild bird mortality also was seen elsewhere in the Netherlands. The highest weekly mortality was in the second week of November, when about 1,500 dead wild birds were reported, roughly 10 times background levels. But please note that these are raw data and they still need to be corrected for duplication. The main groups that were involved were water birds and raptors, and the main species that were involved included barnacle, greylag and Brent geese, mute swans, Eurasian widgeons, buzzards, and peregrine falcons. High-path avian influenza virus of the subtype H5N8, or a reassortant virus H5N1, has been diagnosed in the following water bird species and raptor species in the Netherlands during the first weeks of the outbreak. These data are from the lab of Nancy Berends at Wageningen Bioveterinary Research for dead birds and of Ron Fouché at Erasmus Medical Center for live birds. And no doubt Nancy will provide more details on the phylogenetic analysis of these viruses and perhaps a more updated list. The current measures to deal with high-path avian influenza in poultry in the Netherlands and elsewhere in the European Union, as reiterated recently by EFSA, are influenza surveillance in wild birds and poultry to quickly pick up the virus, high biosecurity of poultry farms to prevent incursion of the virus into the poultry flock, and culling of the whole flock if the virus is nevertheless detected in order to quickly stamp out the infection. Although these methods have been successful to keep poultry largely free of high-path avian influenza in developed countries a few decades ago, they are not sufficient to prevent the disease in the current situation. This is both undesirable and frustrating, and I here suggest a few additional or alternative measures that may help to reduce the risk of high-path avian influenza, as well as having other health benefits. Lowering the production of poultry meat and eggs has been proposed by several authors, mainly for environmental reasons. But it would also reduce the risk of high-path avian influenza, which originates primarily from intensive poultry production. At the same time, consumption also would need to be lowered, and the publication by Poor and Nemecek shows how. Reducing flock density, that is having more space in between flocks, already was mentioned as a measure to reduce farm-to-farm spread and thus limit the size of high-path avian influenza outbreaks. Several studies have shown associations between high-path avian influenza outbreaks on poultry farms and the proximity to water bird habitats and presence of waterfowl, indicating that avoiding such habitats for the establishment of poultry farms would reduce the risk of virus incursion. And finally, preventive vaccination of poultry would both protect them from morbidity and mortality from high-path avian influenza and take away the need for culling. I here summarize the One Health impact of high-path avian influenza and intensive poultry production, as well as the effects of both current and proposed measures. In short, high-path avian influenza and intensive poultry production in the Netherlands have negative health impacts on poultry, humans, the environment, and wild birds. The current measures have a positive effect because the risk of high-path avian influenza is reduced by surveillance and biosecurity. However, culling has a negative health impact on the culled poultry, as well as increasing the risk of high-path avian influenza for those people who are involved in the culling activities. Furthermore, the need to restock poultry farms after culling comes with an additional impact on the environment. The proposed additional or alternative measures have positive effects on the health of all sectors by reducing the risk of high-path avian influenza, by taking away the need for large-scale culling, by lowering the emission of particulate matter and greenhouse gases, and by reducing the area of land that is required to produce poultry feed. In conclusion, I have shown that there has been a marked growth in poultry consumption and production in the EU, and especially in the Netherlands, and that large-scale commercial farms are the primary source of high-path avian influenza virus conversion events. Both this virus and the intensive poultry industry have negative health impacts on people, poultry, wild birds, and the environment. The impact on wild birds in the current outbreak is illustrated by the high mortality of raptors and water birds, including barnacle geese and red knots. The current measures are inadequate to prevent these outbreaks, and I propose additional measures which may have substantial health benefits for people, animals, and the environment. Evaluating and implementing such additional measures successfully may not only improve the One Health situation in the Netherlands, but also provide a positive example for other countries. Thank you for your attention.
Thank you, Professor Thijs Kuiken, for your very interesting presentation. Some of your stats were absolutely flabbergasting. 25% land use by chickens in our country. I mean, that's the same percentage as urbanization in Holland. That's just incredible.
Yeah, it's a big industry.
Yeah, it is huge. You said there was only positive effects of the proposed prevention measures, but you didn't take into account economic situation and job loss. Is that why didn't you incorporate it?
I was concentrating on health aspects, and indeed, there are 22,000 people working in the poultry industry, most of them in meat processing and in providing feed, sheds, and mechanical equipment. And I also indicated that if we made changes in health policies, we need to take this into account and make sure that the livelihoods of these people are considered. Yeah. It is possible that by in changing the way that things are done, there would be employment gained in other ways, and that would possibly help these people.
Yeah, great. I have a few questions for you from the chat. One is from Joukje Siebenga. She's wondering whether the cause for the increase of reported dead wild birds partially lie in the fact that many more people now spend time walking in nature due to the COVID-19 restrictions, and therefore they're finding more dead birds. Do you have an opinion on that?
Yeah, that's a very good point. We always have to take into consideration what the observer effort is. And in this, the raw data that I showed you, this has not been done so far. I did indicate that the mortality observed in the peak week was about roughly 10 times higher than seen during background mortality. But we will need to correct the mortality rates for the background mortality that is taking place, and also for observer effort based on what people are seeing in times where the outbreak is not taking place.
Yeah, this is true. And then we have another question from, or remark from S.J. de Groot. In addition to the Netherlands implemented a rapid alert system and keeping poultry indoors during outbreaks in the wild population. So it's basically both. So the rapid alert system, of course, you get more reports, but then keeping poultry indoors, that would be a reduction, of course, hopefully.
Yeah, it's clear that this is a part of improved biosecurity by keeping poultry indoors, and clearly it does help if given the fact that highly pathogenic avian influenza virus has been widespread in wild birds in the Netherlands. It shows how well biosecurity has worked, that only a small number of locations the virus has entered into poultry farms. So certainly it helps, but it doesn't prevent the incursion of virus onto poultry farms, as well as the occurrence of the disease in wild birds completely.
Yeah, okay. Let's do one more question, and then please keep asking questions in the chat. We'll use them for the panel discussion after the second talk by Nancy Berends. But there's one more question that I promised to ask. So, Professor, let's see, it goes quick now. What's going on at the moment? Are you still observing outbreaks in wild birds now? But I think actually that Dr. Nancy Berends is going to talk about that. Am I right?
I'm sure she will, yes.
Okay, so let me hold the rest of the questions for the panel discussion. Professor Thijs Kuijens, for now, thank you very much. We are now going to switch to Dr. Nancy Berends. She's a virologist and head of the National Reference Laboratory for Avian Influenza and Newcastle disease at Wageningen Bioveterinary Research. She's responsible for all diagnostics regarding these animal diseases and is ready 24/7 to conduct diagnostics of notifiable animal diseases such as avian flu. She's also part of the expert group Animal Diseases that advises the government on animal disease threats. The title of her presentation for today is Introduction of Highly Pathogenic Avian Influenza Viruses in the Netherlands in 2020. Dr. Nancy Berends, the floor is all yours.
Thank you for your kind introduction.
Yeah, we have your slide.
Yes, thank you. So today I would like to tell you a bit more on the outbreak that we are currently having in the Netherlands of highly pathogenic H5N8 viruses. So avian influenza is an influenza A virus. Its genome consists of 8 segments, RNA segments. And on the outside of the virus particle, you will find the hemagglutinin and neuraminidase proteins. These determine the subtype of the virus. There are 16 different H types and 9 different N types. So this results in a subtype as, for instance, H5N8, as we are experiencing now. Low pathogenic viruses, they circulate in wild water birds, which are the natural host of these viruses. But they can be introduced into poultry by infected feces. In poultry, subtypes H5 and H7, they can develop into highly pathogenic viruses. And this has happened in the past in Asia, where this specific lineage of H5N1 viruses, there were many outbreaks in poultry, and then there was spillover of this virus back into the wild bird population. So now wild birds can also carry highly pathogenic viruses, and this is a new situation. We currently experienced several outbreaks in the last years, and today I would like to talk about the outbreak of 2020, where an H5N8 virus was introduced. It infected wild birds, also poultry farms and hobby holdings. So what is the difference between low and high pathogenic viruses? Low pathogenic viruses cause no or only mild disease symptoms in chickens. They may reduce egg productions or, for instance, food and water intake. And that is because these viruses can only replicate in the respiratory or intestinal tract of the virus. However, when there is a mutation in the HA protein in the cleavage site, this can change the pathogenicity of the virus. This then causes severe disease symptoms with up to 100% mortality of chickens. And that is because the virus then can replicate in the whole body of the chicken, which is called systemic replication. So the pathogenicity of a virus is determined by an experiment in animals. It's an intravenous injection of the virus into chickens. And when the score is above 1.2, it means it's a highly pathogenic virus. And the pathogenicity in chickens has no relation to the pathogenicity for humans. So like I just said, only H5 and H7 subtype viruses can mutate to high pathogenicity. And that's why these are notifiable animal diseases. When there is a suspicion on the farm, samples are taken by the Dutch Food Safety Authorities, the NVWA. And these are then sent to our institute. We are the reference laboratory for avian influenza. The samples then are first tested by mPCR, which is a general PCR that detects all influenza A viruses, and then are tested by subtype-specific PCRs to see if they are H5 or H7, so notifiable diseases. This is a 24/7 service of our institute because rapid testing, of course, is required to prevent virus spread. The consequences of an outbreak in poultry are large. That was also discussed by Thijs. The farm is cold, as you can see in the top pictures. It also means that there will be screening of the farms in the 3 kilometers around the infected farm. And there will be limits on transport of chickens and poultry products within a zone of 10 kilometers around the farm. So large implications. The viruses thus now are spread by migration of wild water birds. The birds we have in the Netherlands in the winter, they come from breeding sites in Siberia, in Russia, where they spend their summer holidays. They gather together and they spread viruses. When they fly to Europe and to the Netherlands, they can take along these viruses and then introduce them into the Netherlands and into poultry farms. This particular year, in 2020, there was no mortality reporting at these wild bird breeding sites in Russia. However, in the southern part of Russia, near the border with Kazakhstan, there were highly pathogenic H5 and 8 viruses detected. These are not the areas where there are large groups of birds gathering for the summer, but there are some species that are known to migrate between these areas also and Europe. So we were a bit worried, and we followed the signals in countries east of the Netherlands. So these birds will fly along flyways and will first come to countries such as Poland and Germany. But none viruses were detected until suddenly on October 17, 2 mute swans were found in Kokanje, which is in the central part of the Netherlands. They were submitted to testing, and we found a highly pathogenic H5 and H virus in these 2 dead swans. And this was so the first detection of this highly pathogenic virus in Europe in the fall of 2020. This also shows the importance of the Dead Wild Bird Surveillance Program, which is a program where there are 3 or more dead water birds at one location found. They can be submitted for testing. And you can see that this serves as a very good early warning signal for the poultry sector, because at that time we knew already that there was virus around. The numbers of wild birds submitted were large this year. I can show you an overview. In October, we received around 50 dead birds, and about 50% of them tested positive for the virus, shown in red here. In November, there was an enormous amount of birds found dead. We received around 300 birds, and half of them tested positive. Then in December, the numbers were lower again. Around 100 birds were submitted for testing. And you can see here that also the number of positive birds is declining. There's only around 10% positive. And I think the numbers in January will be similar. So around 10% of the birds we receive test positive for the virus. Early in the epidemic, we mainly received swans and species of goose. And later in the epidemic, this changed. We see more birds of prey, probably because they fed on sick or dead water birds. And we also find a different species, so not water birds, migratory species, but other species of birds that were infected. This shows an overview of all the locations where dead wild birds were found and tested positive for the virus. You can find this map on our website. It's a real-time overview. And what you can see here is that the virus was mostly found in birds in coastal areas. We detected in most birds an H5 and 8 virus, but there were some exceptions. In 5 birds, we detected an H5N1 virus, and in 1 bird, there was an H5N5 virus. So also 2 different subtypes were found in this epidemic. As I just discussed, the dead swans were the first. They tested positive in October 20. And actually directly after that, the government decided that all poultry must be confined indoors. And that already happened on October 23. But not, I think it's only 5 days later, we seen the first introduction in commercial poultry, which was in Altforst. So the chickens were not kept outside anymore, but still the virus was able to enter on this farm. To give you an overview of the diagnostics, so there was this notification, a rapid increase in mortality of the chickens on October 28. The samples arrived at our institute late in the afternoon. We directly started the MPCR and the H5 PCRs, and they were positive already the same day, just after midnight. And the next morning, the farm was culled already to prevent spread. And we subtyped the virus as H5 and 8. So we think early diagnostics, or early notification, rapid diagnostics, and culling are very important to prevent spread of highly pathogenic viruses. Sadly, many farms followed. After this first farm in Altforst, 10 farms became infected. Most of them were chicken farms. There was also one farm with ducks and one farm with turkeys. The last infection was this year, January 5th. And most of the subtypes, the farms were infected for H5 and 8 subtypes. With the exception of the farm in Buitenpost, it's number 9 here labeled in yellow. There an H5N1 virus was found. Of course, we were very interested in where did viruses come from, and we performed full genome sequencing for the viruses that were isolated from the index cases in wild birds and from viruses isolated from the poultry farms. And we were trying to answer questions like, what is the origin of the virus we find in the Netherlands, so of this H5N8 virus, but also of this other subtype, this H5N1 virus. And finally, are there indications for transmission between the farms? So did the virus transmit from farm 1 to farm 2, or were they all separate introductions by wild birds? So this is the first analysis that we did. It shows a phylogenetic tree for the HA segment. And in red, you can see all the viruses that were isolated in the Netherlands. And in green, you will see all the viruses that were isolated in Russia and Kazakhstan. And what you can see is that they cluster together. So they're very related. And these viruses all belong to the H5 clade 2344B. A bit more distantly related, we find this virus in Iraq that was found in 2020. Viruses in Egypt that were found before that, 1 year before that. But the virus is also still highly related to the viruses that we saw in Europe and also in the Netherlands in the epidemic of 2016. And finally, this virus is not related to the viruses we found in Eastern Europe and Germany earlier in this year. So in the start of 2020, viruses were found in this area. And the viruses we have now are not derived from Europe. They are derived from Russia. I also like to show you that this H5N1 virus for HA, it clusters within the group of H5N8 viruses. So it's also a clade 2344B virus. We performed this for the HA segment as is shown here, but also for the other gene segments. And we've still found similar results. So in conclusion, we performed a phylogenetic analysis showing that the virus belongs to clade 344B. The genetic constitution of this virus is still very similar to the one that we saw in 2016. And we show that's not related to Eastern European or German viruses that were found early in 2020. So in this picture, we try to summarize this. You can see here the viruses that are found in Egypt the year before and in Iraq were found later in Russia and Kazakhstan. We don't know which birds were involved, which one birds carried these viruses to southern Russia. So these are not the breeding sites, but specific species are known to fly from these areas to Europe. And then it moved on further into Europe from the Netherlands. So the virus was likely introduced by migratory birds from this area in Russia and Kazakhstan. We know that the first birds we found were mute swans. They do not migrate from this area. But also in Cocaigne was found a dead Eurasian widgeon, and this is a kind of species that does migrate from this area in Russia. So this could well be that, for instance, Eurasian widgeons have been involved in carrying the viruses and introduction into the Netherlands. Then I like to focus a bit on the other subtype we found, which was an H5N1 virus. This is of particular interest because H5N1 viruses are known to be associated with human infections. This is a report I took from the last situation update from the WHO. It shows that these viruses have been found specifically in earlier years in many countries. And they also resulted in around 50% mortality rate. So we were very interested to know if there would be a human risk of the H5N1 that we find now in the Netherlands. We performed genetic analysis and find no zoonotic features. And also phylogenetic analysis shows that it's not related to human viruses. And I show that in this tree. Again, a phylogenetic analysis of the HA segment. On top
We seem to have a little technical difficulty. Nancy, maybe you can hear me, but we can't hear you anymore and you are frozen. So I think we'll just wait a second before Nancy comes back. In the meantime, questions have been pouring in, so I can ask Thijs Kuiken. I can ask you a few questions. Let's see. Thijs, you still there?
Yes, sure.
Okay. Let's see. It's one question from Paula Polman. Do your additional measures have any influence on wild birds with highly pathogenic avian influenza coming from Asia?
So they don't have any influence on the current situation with wild birds because that involves highly pathogenic avian influenza virus of the subtype H5 that originated in Asia and is now more and more being carried by wild birds and also being transported to Europe. But it does reduce the risk of new spillovers like this happening from the Dutch poultry industry. For example, as occurred in 2003, when there was such a conversion event of low path to high path avian influenza virus in Dutch poultry, and the virus also spilled over to a number of wild birds in the Netherlands. So of course, you have to start somewhere. So the suggestions are for these measures to be taken in the Netherlands, but hopefully also other countries would follow suit.
Yes, I agree with that. Hopefully they do. Another one from Claire Guinat. Great talk, thanks. I have a question related to one of your aims, which, if I understand well, is about inferring the wild bird mortality in in percentage of the population. She's just wondering, how do you plan to make such type of inference?
Yeah, this is a multidisciplinary effort, and we have in the consortium many expert ornithologists, and they are making a lot of observations of wild birds in the Netherlands as well as in other countries, and therefore they have quite a good idea of the numbers of wild birds in the Netherlands, and by having an estimate of the wild bird mortality, we can then relate it to the estimated wild bird populations in the Netherlands during this winter.
Can you also see if the population of wild birds is then stable, or is it increasing or decreasing?
These calculations or these observations are done every year. So indeed, people can see whether wild bird populations are decreasing, stable, or increasing. And this is one way, if the mortality is very high, and this has been seen, for example, for another infectious disease, Trichomonas infection in greenfinches, it's been seen that actual observed greenfinch populations have decreased in number as a result of this infectious disease.
Do we have greenfinches in Holland?
Definitely. They're widespread in the Netherlands. Yeah, and they also suffer from Trichomonas infection, and there has been mortality seen.
They sound very exotic, but I'll look up what they look like. Thank you.
Groenling in Dutch. Yeah, in Latin, chloris, chloris, beautiful name.
That is really remarkable. Yes, very nice. I definitely have to look it up now. Another question regarding vaccination strategy. Should highly pathogenic avian influenza vaccination strategy require a system to quickly adapt to circulating variants similar to human?
Probably there does need to be such a system, yes, indeed. However, it's not. It may be that the change in the viruses is not so fast as it is in humans. In humans, every year the virus is checked for changes, and the vaccine is adapted. In the last few years, there's been an incursion of, as Nancy said, this clade 2344B. So a vaccine based on such a virus would be suitable for protecting poultry from infection.
That's good news, but I believe it wasn't incorporated because it results in limited export possibilities, right?
Clearly, yes. There are European rules that prohibit vaccination at the moment of commercial poultry farms. Yeah. This would need to change if we want to remain able to export poultry products from the Netherlands.
Yeah. Well, thank you, Thijs. Nancy, I see you're back. We lost you.
Yeah, apparently.
Yes, but we're very happy to have you back.
Me too.
Are you okay taking over where you left?
Yeah, I will try, but I need to upload the presentation again. Just wait.
No, that's not a problem. Yeah, we can see the presentation.
You can see the presentation.
Yeah.
I will try, and hopefully I will stay in now.
Fingers crossed. All right, floor is all yours.
So we were talking about this H5N1 virus that caused human infections. And we were wondering about the origin. And this is, I just showed the virus is not related to the H5N1 human viruses. We think the virus just arose from this H5N8 highly pathogenic virus, as you can see here in green. When two viruses affect the same host, they can exchange genetic information in a process that is called reassortment. And we think that is what has happened here. So in green, you can see here the highly pathogenic H5N8 virus. And in blue, you can see a low pathogenic virus. When they infect the same host, they can result in a new virus, such as, for instance, this highly pathogenic H5N1 virus that contains both elements from the green virus and elements from the blue virus. So you have this new virus, but it's still related to this H5N8 virus that causes the current epidemic. And so it's not related to human H5N1 viruses. Finally, we studied whether there was a connection or between the farms, if the virus could have been transmitted between these 10 farms that became infected. And therefore, we used genetic information to analyze the relationship between these farms. We saw that there are nucleotide differences between the viruses that we found at these farms. For most of the farms, except for the two farms in Altforst and Puifleek, you can see them here. They're located within a 3-kilometer distance with 6 days just between the two outbreaks. So there's a possibility that the virus was transmitted between the farms. Although you can never exclude that the same bird visited these two farms and they got infected by the same wild bird virus this way. But for the other farms, we can be sure that these are separate introductions from wild birds. I like to end with a bit on pathogenicity of these viruses for wild bird species, as I just showed that the H5N8 viruses found this year and in 2016 have a similar genetic constitution. We saw that both viruses indeed cause high mortality in the wild bird population, as was also just discussed by Thijs. But in 2016, we found mostly duck species deaths, such as Eurasian widgeons and tufted ducks. Whereas now we see mostly species of geese being affected. We performed an experiment in chickens first to study the pathogenicity of the virus. And this is an experiment in which 10 chickens are inoculated with the virus. And then they're monitored for 10 days. And every day the chickens are scored whether they appear normal and they have a score of 0, then they are sick 1, very sick 2, or whether they are dead. And then they receive a score of 3. And this means that the maximum score in this assay is 3.0 when all your chickens would die on day 1. And we can see that for actually the viruses from all the different epidemics, this value is very close to the maximum score of 3. Also for this virus that we found this year. When we performed the same experiment in ducks, we do see differences. And you can see here that the IFPI score for the H5N8 2020 virus is a lot lower. It's 1.7. And this is not the same as we find for the 2016 virus. And we were a bit surprised by that because these viruses are very similar. They have a similar genetic constitution. Although when you look into nature, we think it's in agreement with the limited mortality we see in wild ducks. So mostly geese species are affected now. And of course, very interesting research questions are now which factors cause these differences in pathogenicity. So are there specific genetic features that determine the pathogenicity for specific host species? Or is this more in the wild bird population? And you can think of co-infections with other pathogens or pre-existing immunity in specific wild bird species. And I like to end with the One Health involvement of the veterinary labs. There's this OFFLU network, which is an animal health organization network in which it collects information from all the veterinary laboratories. So information on pathogenicity of wild birds, also on mortality, but also on viral sequences that are found. The analysis is performed to study the evolution and the antigenic changes that we see in the viruses in the different countries. And this showed actually that there are no changes still in this H5 clade 2344B. We just had this meeting with the steering group showing that there are no indications for increased zoonotic potential or changes in the virus. And this is also the input for the WHO human vaccine composition meeting that will be held in February, in which there will be a review again of the need to develop new vaccines and select strains for these vaccines. But we must remember that avian influenza viruses evolve quickly due to their high mutation rate, but also because they can acquire rapidly new features due to this reassortment process. So I think it's very important to always closely monitor avian influenza viruses for pandemic preparedness. And I like to end by thanking all the people in the avian influenza group that were involved in this research, but also all the people that were involved in diagnostics during this outbreak. We received many samples and tested during nights and in weekends. So I like to thank them all for their efforts, and this outbreak is still ongoing. And of course, I also like to thank you for your attention. Thank you.
Thank you, Dr. Nancy Berends, for coming back to us and continuing your presentation. Your timing is immaculate because we're now out of time, so perfect. I will ask one more question from the chat function. What to do when you find an injured or dead bird? Normally they would take it to the animal ambulance, but what should people do?
Yeah, you should, of course, take care that you do not get into contact with infected dead birds, because there's always a risk of getting infected, and the first human infection could cause this new pandemic. And also you should be taking care of where to bring this bird to, because if you bring it to an animal shelter where there are lots of other wild birds, these could infect the whole facility. So care must be taken. They need to be put in quarantine, these birds.
Yes, okay. Well, thank you both for your very interesting presentations. To the audience, you can still submit your questions in the chat function. We will submit them to the authors and find a way to get the questions either in the video that we'll post on YouTube later or to our NCOH website. We'll find a way. Both Professor Thijs Kuijkens and Dr. Nancy Beerens, thank you very much. NCOH communication team, thank you very much for organizing this webinar. The next webinar will be on the 4th Thursday in February, and the topic will be fighting COVID-19 in cats and dogs. If you have any questions or comments, you can reach us on info@ncoh.nl. And you can also send us suggestions to organize webinars on topics you find interesting. So thank you for dialing in and see you next month. And that's it. Thank you.